Good morning everyone. Welcome to all 4C learning classes. This is session 11. Let's have some quick look of the digestion in the stomach. Earlier we have discussed the digestion in the oral cavity when the food added by the saliva. The saliva, which includes the mucus and salivary amylase as well as the water and salts. The tongue, which pulverizes the food and forms the food bolus, where the salivary amylase ptyalin act upon the starch. Starch of 30% in the oral cavity is digested. The digestion in the oral cavity of the carbohydrates only, which means our process of digestion begins with the starch or carbohydrates. So 30% of the starch is digested. This digested food gradually move into through the pharynx, esophagus, reach the stomach. The digestion in the stomach. The digested part, partly digested part, food in the buccal cavity, oral cavity is the food bolus, which gradually move on to the stomach. In the stomach, as soon as the food reaches the stomach, the gastrin is a hormone released and it stimulates the secretion of the gastric juice. The gastric juice, which is highly acidic. Gastric juice is highly acidic, which includes the inactive enzymes pepsinogen, prorenin, and it is with the caseal intrinsic factor. It is with mucus and the gastric juice, which is highly acidic because of the HCl. The role of the HCl, once the food reaches into the stomach, HCl kills the bacteria. HCl is an enzyme activator in the acidic medium, which convert the pepsinogen into pepsin, prorenin into renin. Remind you one thing, renin, when it is pronounced same like RE, renin action is in two cases. One is in the digestive activity and another in the excretory function, also osmoregulations. Here it is RENNIN, whereas there RENIN. So the inactive enzymes are converted into active enzymes by HCl. Then the pepsinogen into pepsin and the prorenin into renin. Pepsin, once it get activated, it acts upon the proteins and which require acidic conditions. That's the reason why the stomach is the part where proteins are digested. Since the action of the pepsin is in acidic condition, however, gastric lipase is also present. Lipase is the fat digestive, but the acidic condition inhibits the digestion of the fat. One point, the reason for the non-availability of the fat digestion in the stomach is one, acidic condition. Second is the food is the fat is not emulsified. Emulsification is done by means of bile juice which contain the bile salts, which are not available. This is the reason why the digestion of the fat is inhibited. However, gastric lipase is present. Next, in the infants we have discussed the prorenin is present. Prorenin is seen in only the infants, which is converted into renin by HCl. The infants, now the thing is, the renin is RENNIN is a renin seen in only in children, not in the adults. The due reason is renin prevents the flow of the milk as it is from the stomach into the intestine. That is, renin act upon the casein protein and convert it into paracasein. Paracasein, in presence of calcium ions, it is converted into calcium paracaseinate. Now, in the stomach, the pepsin act upon calcium paracaseinate and convert it into peptones. And renin is what commercially synthesized from cough gastric mucosa. This is the important point to remember. That is in the form of renite tablets. Renite tablets. Renite tablet. Commercially produced from cough gastric mucosa. That is it is extracted from cough gastric mucosa and commercially prepared, which is given to the infants where there is a deficiency of the renin. Next, in the stomach, this partly digested food, which is said to be the chyme, the partly digested acidic food is what named to be the chyme. This partly digested food gradually moves from stomach into the intestine, which is regulated by the pyloric sphincter. The point to remember here is certain times the food, the partly digested the food of the chyme, which is pushed into the esophagus from the stomach. This causes the burning sensation, which is said to be the heartburn or pyrosis. Heartburn, and it is named to be the what is called the burning sensation in the gut, that is particularly in the stomach and esophagus region. It is nothing to do with it is a hyperacidity also we consider. Hyperacidity or the ulcer. The due reason is, that is the case with the stomach, the gases that are produced, that is the stomach which contain the digested food is pushed into the esophagus. This causes the heartburn or the hyperacidity. It is nothing to do with the heart problem. It is nothing to pose the problem with the heart, but it is a digestive problem, which is ulcers. It is the regurgitation of the food from stomach into the esophagus. This generates the what is called heartburn. It is nothing to do with the heart problems. Next, even the food that is present in the stomach points to remember that mucus prevents the excoriation of the gut. Excoriation literally means excoriation. In Latin, it is said to be skin off, and the corium means the skin. That is the mucus which prevents the excoriation of the action of the digestive juices, particularly the HCl on the gut. Moreover, the gut is protected from the what is called autodigestion. The autodigestion means digesting its own parts. Basically, the cells are made up of the proteins, which may be damaged by proteases enzyme or proteinases. That is the how it is avoided. It is nothing but the enzymes released in an inactive form. So gut protected from excoriation by mucus secreted from the goblet cells, and also the enzymes released in an inactive form, particularly the pepsinogen and prorenin are inactive enzyme. The inactive production of enzyme avoids the extracellular digestion or cellular digestion of the body parts, and they are released in inactive form and are converted into active. The pepsinogen by means of HCl converted into active pepsin. Then at the site of the digestion in the gut, it is act and convert the proteins into the partly digested peptones, proteoses and the peptides. This partly digested food in the stomach is what name it to be the chyme. This is what we see. Next, we move on to the digestion in intestine. Digestion in intestine. Digestion in intestine. The partly digested food that reaches into the intestine. Intestine, small intestine. Small intestine 6.25 meters. Includes duodenum C-shaped, then the jejunum and ileum. In this connection, we learn the digestion is completely takes place in the small intestine, and most of the digestion is in the duodenum. Let's see how we summarize such a process of digestion in the intestine, which is as follows. Once the food chyme enters into the small intestine through the pylorus opening, which is guarded by pyloric sphincter. Pyloric sphincter regulates the flow of the food from stomach into the duodenum. Once the food reaches into the duodenum, the changes we find is vagus nerve is said to be. Vagus nerve is the 10th cranial nerve which stimulates the secretion of the secretin hormone. Secretin hormone is the one which causes the secretion of bicarbonate from pancreatic juice and also the juices, bicarbonates. Why? Generally, the food that is traveling from stomach into the duodenum is acidic. It is to be neutralized. The neutralization takes place by bicarbonates. So, the food that when enter into the intestine, the stimulus by cranial nerve, 10th cranial nerve vagus, stimulate the secretion of secretin, which stimulate the what is called release of the bicarbonates of the pancreatic juice and the water. This causes the neutralization of acidic effect of the stomach. And then even we see the hormonal influence secrete the release of the juices, digestive juices into the intestine. There are three we have seen earlier. The action of the digestion process involved with the digestive juices, which includes the salivary juice, gastric juice, bile juice, pancreatic juice, intestinal juice. So far we have discussed the action of the salivary juice in the oral cavity, action of the gastric juice in the stomach. Now it is the remaining three, which is the bile juice, next the pancreatic juice, next the intestinal juice, or it is the saccus entericus. The complete digestion in the intestine, small intestine, takes place where alkaline condition exists. Alkaline condition is required for fat digestion, earlier we have seen. And the stimulus by the neuron as well as the hormone causes the release of the bile juice, pancreatic juice, intestinal juice. Bile juice is and pancreatic juice. Earlier we have discussed. Hepatopancreatic duct or ampulla of water releases. This is a combined duct of the common bile duct with that of the main pancreatic duct joined together form the hepatopancreatic duct or the ampulla of water. which releases the bile juice and pancreatic juice. Both are alkaline. So this adds to the alkaline condition in the intestine. Bile juice is an enzymeless secretion. However, the bile juice is enzymeless secretion. It play a role in digestion. How? The emulsification. Until and unless emulsification takes place, fat digestion is not initiated. This is the reason why in stomach the fats are not digested. However, gastric lipase is present, and acidic condition is one more reason. Bile juice include bile pigments, bilirubin, biliverdin, which are excreted outside, and the bile salt for emulsification of fats. That's the role of the bile juice in the intestine. Next, pancreatic juice. Pancreatic juice is also alkaline in condition, and it includes the protein digesting enzyme, carbohydrate digesting enzyme, fat digesting, as well as the nucleic acid digesting enzymes. That is trypsinogen, chymotrypsinogen, carboxypeptidase, or the protein digesting enzymes of the pancreatic juice. Next, the fat digesting pancreatic lipase, which is said to be the steopsin, and fat digesting lipase pancreatic nucleic acid digesting nucleases, DNAs and RNAs are present. Next, the intestinal juice, saccus entericus. Saccus entericus or intestinal juice secreted from the crypts of liver cone. Generally, combination of crypts of liver cone and Brunner's glands is said to be the intestinal juice. It is an alkaline condition itself. This intestinal juice contains enterokinase, an enzyme activator. Earlier we have seen some enzymes are released in an inactive form whose activation is needed. In the gastric juice, the HCl is an enzyme activator. In the same way, intestinal juice, saccus entericus, enterokinase is an enzyme activator. Enterokinase. Next, the disaccharidases. In the case of the pancreatic juice and earlier in the oral cavity, the carbohydrates are converted into what is called disaccharides. Disaccharides in the sense maltose, sucrose, lactose, and alpha dextrins, isomaltose, and elastin are what is called present as a disaccharides. However, elastin is nothing to do with the what is called the carbohydrates, but it is seen. That is in the intestinal region. What I am saying is, and besides that, the intestinal juice contain the enzymes named to be nucleotidases, nucleosidases. These are the nucleic acid digesting enzymes. So collectively, the alkaline juices are released into the duodenum. That is, hepatopancreatic duct brings the bile juice and the pancreatic juice. And the intestinal juice or saccus entericus, which is the combination of crypt of liver cone and Brunner's glands, are releasing their secretions into the duodenum. Most of the food digestion takes place in the duodenum. Then it gets absorbed in the ileum, which is a characteristic feature with the villi. So first of all, the action of protein digestion, will see first protein digestion, as the food includes the proteins, carbohydrates and fats. So first, the protein digestion. Protein digestion is by the enzymes of both the pancreatic juice and intestinal juice. The list of the enzymes of the protein digesting are in the intestinal region. It includes now in the intestine protein digestion. Protein digestion involves the enzymes called of the pancreatic juice, which includes carboxypeptidase, next the trypsin, chymotrypsin, and intestinal juice, aminopeptidase, dipeptidase. These are the enzymes acting upon proteins. So, the enzyme protein digesting, name it to be the proteinases or proteases. In the case of the intestine, protein digestion involves the proteases are nothing but, first of all, we'll discuss the action of the pancreatic juice. The protein digestion involves the action of the pancreatic juice. Pancreatic juice contain inactive protein enzymes such as trypsinogen, chymotrypsinogen and procarboxypeptidase. Earlier we have seen, recall your memory, there are the enzymes are released in inactive form. The protein digesting inactive form in the intestine include trypsinogen, chymotrypsinogen, procarboxypeptidase. These are the inactive enzymes of the pancreatic juice. Now they require the activation. This activation is done first done by means of an enzyme activator, name it to be the enterokinase. Enterokinase is an enzyme activator of intestinal juice. So what we come to know, it is a combined action. First the activation is needed. So enterokinase is of intestinal juice, an enzyme activator of alkaline condition. Enterokinase converts the trypsinogen into inactive form into active trypsin. And the point to remember, as we discussed at the stomach, autocatalysis, auto self catalysis, breaking. That is, the trypsin once become active, this trypsin again act upon its own inactive form called trypsinogen and convert it into active trypsin. So this is autocatalysis. The action of an enzyme in its own inactive form and converting it into active form. So it is a autocatalytic enzyme. So let me ask you a question. Autocatalytic enzyme in the acidic condition is, or else yes, autocatalytic enzyme in acidic condition is pepsin. Pepsin, once it become activated, act upon its own pepsinogen and convert it into active pepsin. And autocatalytic enzyme under the alkaline condition is the trypsin. Trypsin act upon trypsinogen and convert it into active trypsin. So 4 points to remember. 1. Enzyme activator in acidic condition is HCl. Enzyme activator in intestine is enterokinase. The autocatalytic enzyme in acidic condition pepsin, and autocatalytic enzyme in the alkaline condition is trypsin. The trypsin even furtherly act chymotrypsinogen and converted into chymotrypsin. Then even procarboxypeptidase is converted into carboxypeptidase. So this is all what done by means of trypsin. Even proelastin is an inactive form upon which the trypsin act and convert it into elastin. So this is how the conversion of inactive enzyme into active. Now the active enzymes start acting. That is, the proteins, peptones, and the peptides upon which the trypsin, chymotrypsin, and the carboxypeptidase act and convert them into dipeptides. This witnesses how the protein digestion takes place. Next, the elastin upon which the elastase enzyme what act and convert it into the dipeptides. Next, besides this, we do also find the aminopeptidase act upon aminopeptides. Basically, the carboxypeptides and aminopeptides are said to be the exopeptides outside, whereas the remaining are what's to be the endopeptides, which are present within the amino acid protein. So carboxypeptidase act upon the carboxylic terminal, C terminal of the protein amino acids. In the same way, the aminopeptidase N terminal, N refers to amine, C refers to carboxylic, which are present peripherally. So it is exopeptidases, carboxypeptidase and aminopeptidase. Next, the remaining are breaking the bonds inside, so endopeptidase. This is how the proteins are digested. So proteins, peptones, peptides by action of the trypsin, chymotrypsin, carboxypeptidases into the dipeptides. Again, the aminopeptides upon which the aminopeptidase act. That is, generally the aminopeptidase is what breaks at the amine group, and that results in dipeptides. This is how the proteins are digested. And the products of proteins are not the dipeptides. So, and the products of proteins are amino acids. Now, dipeptidase and enzyme act upon these dipeptides and convert them into amino acids. So, this is how the proteins and the products are amino acids are formed in the intestine. It is the combined action of the pancreatic juice as well as the intestinal juice. Pancreatic juice containing the trypsin, chymotrypsin and carboxypeptidase, whose inactive forms are converted by the action of the enterokinase. In the same way, the dipeptidase and aminopeptidase of intestinal juice furtherly digest. So collectively, the proteins, generally the proteins which are with the peptides and as the first initial stage of the protein digestion from proteins to peptones are formed, then proteoses, then peptide. This is the what is called the products that are produced. That's the reason why we say the proteins, peptones and peptides. Furtherly, dipeptides. So that's the reason why the dipeptidase act upon dipeptides and forms into what is called the amino acids. End products of the proteins are amino acids. This is what we refer to the protein digestion. Protein digestion. Generally, the protein digesting enzyme, if we see the pepsin, which is not seen in the invertebrates, whereas the trypsin is seen.